So guys, now you have learned about the different aspects of the metabolism of nucleotides. Now let's have a bird's eye view of what you have learned, just a final glimpse or a glance of what you have been taught about these different aspects of the metabolism of nucleotides, and also what was actually the significance, especially the clinical significance of this metabolism. So if we, just have this is what we say the concept map. You know that there are two major nucleic acid molecules, DNA and RNA. And of course, the nucleotides of the DNA are the adenine, guanine, cytosine, and thymine, while in the case of RNA, that is adenine, guanine, cytosine, and uracil. So RNA have uracil in place of the thymine. So this is something, it is not new for you. You have been studying this for many years, starting from your middle school, I must say, up till your BS or MS level, and so forth. But it is always good to just to refresh your memory, refresh your what you know about these molecules. So, and I have told you, we have been studying that there are two major types of nucleotides. One is the purine and the other is pyrimidines. And as far as their synthesis was concerned, the purine synthesis was a little bit different from the pyrimidine synthesis, especially in the sense that the purine synthesis started with the ribose 5-phosphate. So you already have a ribose 5-phosphate molecule, a sugar molecule is already there, and then you build upon it the nitrogenous base part of the nucleoside. So this whole process, starting from the ribose 5-phosphate up till you end up with the inosine monophosphate, and this inosine monophosphate then can can give rise to the formation of guanine moieties or the adenine moieties like AMP, ADP, and ATP, or GMP, GTP, and GTP. And of course, then these moieties will become the part of the RNA. But of course, we need DNA, so that is your major information molecule. So these these nucleotides then under the effect of different enzymes, specifically the reductase enzymes, they lead to the formation of deoxyribose sugars. And these nucleotides then are utilized in the formation of the DNA. Fine. You already know that we have been saying it again and again that the major atoms, major structural components of these nucleotides come from specifically the nitrogen group comes from the amino acids, for instance glycine, glutamine, aspartate, and also the N-formyl THF tetrahydrofluorate, while the carbon skeleton is provided by carbonates or carbon dioxide. Then of course this was the synthesis. Then there comes the degradation of purines. The purines, whether it's adenine or whether it's that is AMP, or whether this is guanosine monophosphate, they both converge towards the formation of, during the degradation process, we get the hypoxanthine, which is converted into xanthine, and this xanthine is then changed into uric acid. So uric acid, the final product of the degradation of these nucleotides, and which is this uric acid is then excreted out from the kidneys. Fine. As far as the pyrimidines are concerned, the major difference between pyrimidine and pyrimidine is again in the case of purines, you started with the sugar molecule which is already there, sugar moiety. Here you start with the nitrogenous base, which once formed, then it is added up on as sugar moiety. So this is a difference between the purine and pyrimidine. So you start with the ATP molecule and carbon dioxide and glutamine. All of these provide the major skeleton or major structural components of the nitrogenous base, and you end up with the formation of UDP, which can be, you end up with the formation of UMP, which is converted into UDP, and of course, when you want, it can also be deoxygenated to form DUMP, but you know that DUMP is not the major component of the DNA. So that's why it is not used in the formation of DNA. Then UDP is converted into UTP, which can be converted into CTP. So in this way, you have seen that how the carbon skeleton and the nitrogen components of these nucleotides come from specifically from the amino acids and also from the carbon dioxide. And you also now learn, again you saw or revised that how they differ from each other, the both major types of nucleotides, that is the purine and pyrimidines. Finally, just very shortly, I will like to tell about the different diseases which are involved in the when anything wrong occurs in the metabolism of these nucleotides. One of the major diseases is the gout, especially if there is a problem in the purine degradation. You have this uric acid is increased in your body. This increase in uric acid can either be because of the problem in the excretion of the uric acid, or because of the you get more and more uric acid production. So which means that there is some problem in the metabolism of these nucleotides. So in this way, this metabolism of these nucleotides have a real clinical significance. Another way of seeing these nucleotides, how they are clinically important, is that they are also drug target. For instance, methotrexate is a very important anti-cancer drug. So these and other types of anti-cancer drugs, they are used to inhibit the formation of DNA, and that inhibition in the formation of DNA is because of the inhibition of the purines like TMPs, thymine monophosphate, which is involved in the synthesis of DNA. So once the DNA is inhibited, of course, it also will lead to the inhibition of the cell growth. Specifically, we want the cancer cells growth to be inhibited. So in this way, the metabolism, or especially the degradation process of the nucleotide, is also a target for different types of drugs, especially the drugs which are used to treat cancer. Finally, purines are also involved in the salvage path. Salvage pathway, we have already told you, salvage pathway is that in which you use already made components to make a little bit more furnished component. You don't have to use the whole raw material. You don't have to start from the raw material, from the very crude type of components. You already have, for instance, the DNA or RNA, which when undergoes the degradation, and this DNA and RNA gives rise to, because of the degradation, different bases, nitrogenous bases. This nitrogenous bases then can be used for the formation of the nucleotides like IMP, like GMP, AMP, and so on and so forth. And any deficiency in the formation of these nucleotides during the salvage process also leads to certain diseases like Lesch-Nyhan syndrome. One of the really affects your mental abilities. For example, your cognitive abilities are very much compromised, and self-mutilation. You start harming yourself. अपने आप को नुकसान पहुंचाना शुरू कर देते हैं इस डिजीज के, क्योंकि आपकी mental abilities are not working well. Their cognitive ability are getting failed. And of course, the hyperuricemia, because any defect in the metabolism or degradation of these nucleotides will lead to the formation of more and more uric acid, which can be accumulated, and you get the hyperuricemia. So in this way, you see that not only it is important to know how the nucleotides are synthesized or how they are degraded. It's also very important to know how they are clinically important, how any failure in their metabolism can lead to what sort of disease, and how these different pathways of their metabolism can be target of different diseases, can be target to make drugs for different diseases. For example, one major example is the methotrexate, which is used to treat cancer.